So as Alex said, it's absolutely great to have you here. And our first speaker, we're going to hear about the work that Beatrice Lace has done, but the presentation is going to be given by Thomas Ott. Thomas Ott studied his degree at the University of Göttingen and then went to the University of Manchester for a bit. He did his PhD in Max Planck Institute of Plant Physiology in Gulm, went on to a postdoc in Gulm as well, and then went to Toulouse for a little bit as a Marie Curie Fellow, and then on to the University of Munich, where he had a number of different positions, including an Emmy Noether junior group leader position. And then he has ended up in Freiburg, where he's a professor for cell biology. It's a real privilege to have Thomas here. So I promise I won't fanboy too much, but Thomas has published loads of wonderful papers. I'll mention one of the best papers I've ever read, Jask et al. 2014, I think. I might be showing my age a bit there, but which was all about characterizing remorins in plants. And I remember reading it and being blown away and thinking, this is the kind of science I want to do. So it's really fantastic to have Thomas there. And I won't say any more, but I will hand over to Thomas. So thank you very much, Thomas, when you're ready.
Thanks, Joe. I'm very delighted to be here. In fact, this talk should really be given by Beatrice because she's the one who did the work. Unfortunately, she got sick yesterday, and then we decided to have the seminar for sure that I take over. But she's here, so for the hands-on questions on the technology and developments, really who should get the credit for it. For the work I'm going to talk about, that's, as I said, is Bea's work, and I hope you can all see the screen. So we will kick off the series with a bit of FLIM imaging, and let me just show you a little bit what we are doing in the lab with this, or what Bea is mainly doing. So of course, the general thing is we have a few fluorophores available, but we need to somehow usually spectrally discriminate between them, and then be them in the blue, in the yellow or green or in the red color. But of course, there's many more. And then the problem starts that they're all spectrally overlapping. So we are just very limited in what we can image. And especially in plant cells, we always have this issue of autofluorescence, right? Be it the cell wall, be it the chlorophyll or so, that sometimes really makes imaging, especially of lowly fluorescent proteins, very, very demanding. And it's going as far as that you cannot properly discriminate sometimes whether your signal is a truly derived signal from your fluorophore or from autofluorescence. So this is something that we wanted to challenge a little bit, and it all went hand in hand, I should say, with this FLIM technology being more and more integrated into commercial setups. Yeah, so while it was really a demanding thing and you needed all these extra equipment pieces in earlier times, now it's more integrated into the machines, and that allows you now to not only look spectrally at those fluorophores, but in fact do look at them more by their lifetime. And lifetime means that every fluorophore has this particular, let's call it fingerprint, where we can basically read out a certain lifetime, and explain in a second how this is generated. But in principle, you can basically remember that the lifetime is the average amount of a fluorophore, or the time a fluorophore remains excited before it emits actually the photon. Yeah, and Beatrice, and I have to say she assembled most of these slides here, so thanks very much for this, has put together a few explanatory ones for this one. So what we basically use, and what you need to use usually, is a pulsed laser system, which sends in the picosecond range or so individual laser pulses, and then you excite your fluorophores, and of course you excite your fluorophores, and then the excited electrons at some point return back into the ground state. But at some point the photons are emitted from the fluorophore, and the time in between excitation and this emission of the fluorophore, this is indeed what you record initially. And then you basically get these histograms, you put them all together into a photon arrival time histogram, which you can see here on the right side. Then you fit a curve into this, and this gives you the fluorescence decay profile. And from this, you can then calculate the lifetime. This is how it has classically been done. It works nice. However, it's always this fitting involved, and whoever has worked with the FLIM systems knows that this fitting can be better, can be worse, depending a little bit on your sample, depending how many components you have in there. So we focus we focused in the last years a little bit more on a different mode of analysis, and this is called phasor plot. So the phasor plot is a system where you, of course, rely on the fluorescence decay, but then you do a Fourier transformation of the data and basically plot them along this semicircle. So you bring them in a 2D diagram where they get precise G and S coordinates. So the S and the G here. So you have a position of what we call a phasor cloud. Yeah, and this phasor cloud should represent your fluorophore, and they align along this semicircle. If you have very pure fluorophore and everything, is the fluorophore alone, it sits more or less onto the semicircle, but that can differ, and I'll show you this in a second. Now, the important thing is that what you can do then is you can basically, of course, draw, for example, ROIs around those clouds. And maybe to briefly acknowledge this, the fluorophores, or the fluorescent species, I should say, that exhibit a short lifetime are plotted on the right part of the semicircle. The ones with the longer ones are moved towards the left part of the semicircle. Now, you can now do it either way. So you either go onto your phasor plot and you say, okay, this is an interesting cloud, and I'll show you some original data in a second. Yeah. And you basically take a ROI, look at it, and then find back, and then these pixels are highlighted in your image. But you can also go to your image, yeah, and say, this is my region of interest, and then go back and look at the phasor cloud and the phasor plot and see if you have a single species population or if you have mixed fluorophore populations and so forth. So this is a general mode that is also implemented in some of the systems, and I think we think basically it gives a lot of has a lot of potential for the quantitative analysis of images. And what you would usually get, and that's first depicted as schemes here. So you can, when you basically just have an overall image, you get maybe, let's say, a single fluorescent species and some autofluorescence or whatsoever. Then you have this a single cloud with a maximum, and you have this kind of a bit of fuzzy cloud of data points around. However, of course, when you draw a region of interest, you narrow that down. That is pretty common, of course. So if you now have two fluorophores, they distribute now along this semicircle, yeah, again with the ones with a lower lifetime being positioned to the right and with a longer lifetime being positioned rather to the left, to the other side of the semicircle. And these clouds, as I already said, if they are kind of isolated, then you're talking about kind of pure fluorescent species. However, what of course happens a lot is that you have overlapping fluorescence. So the species or the fluorophores are sitting in the same place. And then you get a line that we call the mixing line. And the mixing line basically connects the two clouds of the pure fluorophores, here S2 and S1, with each other. And then you start to see a cloud forming there. And this is now giving you a mixed population of fluorophores. You can display that differently and highlight different populations by putting these rulers on. But of course, what you also can do again is you draw ROIs around it. And in this ROI, that I think it has a lot of potential for later applications, you can basically see what is your proportion or your stoichiometry of proteins within a certain region of the cell. Yeah, you can say, for example, 60% or 80% in this case derives from fluorophore 1, 20% from fluorophore 2. And that gives you a lot of opportunities now to quantify actually those stoichiometries. And I think this is something we want to continue really working on in the future. Now, to show you some experimental data, what have we done? And we first started with the multiplexing. So Bayer has used a number of different fluorophores, a few from the green, a red, and yellow ones. And you see here the average lifetimes depicted from the literature or from different databases. And then we have tagged those fluorophores towards the different compartments, be it the plasma membrane, the nucleus, the chloroplast, or the endoplasmatic reticulum. And we wanted to ask two questions. So first of all, can we nicely discriminate spectrally overlapping fluorophores due to their lifetime? And how close can those lifetimes be of those fluorophores to be still distinguishable? right? And this, of course, goes hand in hand with this ability to multiplex more and more. So that the smaller the delta in between the lifetimes, the more options you get. So we used, because it's a fast and nice system, Bentamiana leaves and infiltrated them. And for example, then have a nuclear eGFP and a plasma membrane localized now GFP. And that's we did not only those combinations, but in different combinations, either putting them all to different compartments to have fluorophores being of the same color, but spatially separated, but also have fluorophores being at this having the same spectrum or similar spectra, but sitting in the same place. So all of these things we tested, and actually, I have to say it works quite well, and we are pretty fascinated that this gives really new opportunities for imaging in plants. So taking this example, for example, where we took 4 fluorophores, 2 green fluorophores, 2 red fluorophores. The green ones were targeted to the plasma membrane and the chloroplasts, and the red ones were targeted to the nucleus and the ER. For the green ones, we chose now GFP and eGFP. There's a delta of 1.5 nanoseconds in between them. And for the red ones, we chose M scarlet and M cherry with a delta of 2.0 nanoseconds. Now, if you infiltrate these combinations into the leaves, you see this if you go just via normal intensity imaging, so normal spectral imaging. Of course, you cannot discriminate the eGFP and the now GFP, and you cannot discriminate clearly, despite their different localization, of course, the M cherry and the M scarlet. But if you now do FLIM imaging, and this is fast FLIM imaging in the Leica system, you can nicely see that you can do this because now you're not doing the spectral discrimination anymore, but you discriminate by lifetimes. And as I said, the eGFP and the now GFP are having distinct lifetimes. So now we see very nicely that here the chloroplasts are in green, the plasma membrane labeled in red. And similarly, sorry for this. Similarly, we have the ER and the M scarlet, where you can the ER and the nucleus, which you now can very nicely discriminate. So to take this further, we now take this to the phasor plot as I introduced, and now it looks like this. That this is a real phasor plot. This is how they look like. You see that you have 2 maxima being here. You have this indication of this mixing line here, as you see, but of course these are spatially separated, so you get quite nice separated populations. So this one here to the left is the one from now GFP. The one to the right is the one from eGFP. And when you now draw the rows around them, you can see very nicely that you can discriminate. And now you can just split basically the signals. And on the right, we show you that it's now purely the plasma membrane for the now GFP, purely the chloroplast for the eGFP, and here the nucleus for the M scarlet and the ER for the M cherry protein. So meaning that multiplexing for proteins and plant cells is actually very feasible and gets straightforward. The nice thing is it's implemented into the system. Is you have to get a bit into this. So all the getting used to the system is fine, but it really opens up absolutely new opportunities. And if we would have shown you this with, let's say, a tomato root system or whatever, where there's a lot of autofluorescence, you don't care anymore because you basically can take this out of the image on a not with a post-image processing, but on solid data where you say everything, let's say, with a lifetime below 1 nanosecond or 0.5 nanoseconds, you just take it to a different channel and then you can either use it or omit it from your image. So this, I think, is really something that we hope really gets pushed in plant cells, because it provides a lot of opportunities, not only for doing multiplexing, but really maybe to revisit some data and look if the signals, and we've done that also for quite some of our data, look if the signals are truly purely derived from a fluorophore, or if there's also autofluorescent signals in there. And I'll show you an example in a second. So now one approach we also wanted to do is that we take these 2 fluorophores here, M citrine and Venus, which only have a delta of 0.2 nanoseconds. Yeah, so that's very close. So they're spectrally overlapping, and also their lifetimes are now very close to each other. We target them either to the nucleus or to the plasma membrane. And in this setup, you can still discriminate between the two. Yeah, so if you have two distinct clouds, if you roll them, you see very nicely that you can still discriminate them. There is, however, a little bit bleed through. So it starts to get tricky, but it shows that there is really a lot of potential to multiplex if you're having the system under control and somehow know your samples. Because, and we show this, we report this in the hopefully very soon released preprint, that also we have some organ-specific lifetime changes. All right, so another thing that Bea did was then to say, okay, classical situation, you infiltrate two proteins, and of course, now we take two overlapping or spectrally overlapping proteins, so two green ones, again, now GFP and eGFP, and we both placed them in the plasma membrane. We looked at the surface, and no nanodomains this time, Joe. Sorry for that. Yeah, so we looked at the surface, and then we looked, of course, at a secant plane. And what you now can do, because you can now easily discriminate, you see basically that proteins are differently distributed. So you see the mixing again. You see that there's protein populations that are completely mixed, where there's stoichiometry between now GFP, and I maybe can also switch to this one already. Yeah, where now GFP and eGFP are more mixed. Other cells contain more of one and the other protein. And that starts to give you this very nice pattern. And you can start working and see, for example, if two proteins are in the same compartment, how or do we induce cellular changes or whatever your scientific question is. So just saying that behind these fluorophores, using FLIM, there's a lot of information one can make use of, and I think where we can gain a lot of cool science actually, also in plant cells. Now, this has been all done with fluorophores, and this is nice because the teaser image of this series is actually a FLIM image, right, from Johannes Theodorou. We saw that in compressed wood, yeah. And this is nice because, of course, wood, but also other cellular compartments or molecules have autofluorescence. Yeah, so if you just look at autofluorescence, this is a cross-section through a tomato root, you see something. So you see here the Casparian strip, depends on where you cut it. Sometimes here it's elongated, sometimes it's the little dot. You see the exodermis, you see the xylem structures. And this is just autofluorescence. You can see that with a lot of systems, no problem, but it looks all very similar. You can, of course, then do stainings, like basic fuchsin, right, and stain lignin, for example. And you see it, but still it looks very homogeneous. And I think where there is a lot of potential for FLIM is when you now look at this same thing with FLIM, you see that the lignin composition is actually not the same. Yeah, so you get different lifetimes in different cell types, different structures, indicating that there are differences. So FLIM doesn't allow us to tell what exactly those differences are. Yeah, but this is something where probably if you get more and more insights into your process, you will learn about this. But you see that there's heterogeneity in the cell system. And again, I think we can use autofluorescence a lot to do this. So this is the nice part of autofluorescence. The ugly part of autofluorescence is if it interferes with your signal, right? So if you want to see something, and we working a lot on root hairs or on infection in root hairs in a symbiotic context, and this is some work Bea has published some years ago, where we looked at this infection process where rhizobia, so soil-borne bacteria, infect legume plants through this tunnel. And grow, and what you have is the formation of something that we call an infectosome. Is probably a condensate structure sitting at the tip of this, what we call infection thread, at the nucleus and a little bit in between. So it's little dots floating around. And there's a lot of little dots floating around in the cell if you look carefully. But of course, what you also have is a root hair. And when you just look in a curled root hair where the cell walls are really compressed a lot to each other, you see a lot of fluorescence. And this is a protein called vaporin, but doesn't matter here at all. You see basically very nicely the outline of a root hair. You have very strong fluorescence here at the tip. You have some fluorescence here in the middle. And in the end, from intensity plots, you cannot see really what your fluorescent signal is. But when you use FLIM, you suddenly see that all this blue here is all autofluorescence. That's all the cell wall. And your true signal is only this little spot here. Yeah, so again, it's all information is in the image, and you can retrieve it from there. And we think really that this has a lot of potential for imaging. So as one part that we've done already is to use it now a little bit to look more in detail. And this goes a little bit, and the reason why I'm talking about this in the last few minutes is where do we want to go with FLIM imaging? I think we should, of course, increase resolution and get more information. And it's based, and I'll show you the examples for infection because this is our home base in a way. So again, in this paper from Bayer, we showed when we used here this BF05P2 reporter, so a lipid species reporter, that indeed with FLIM we could very nicely show that there's an enrichment of BF05P2 at the tip of this infection structure here, yeah. And also on a side here at the membrane where suddenly we have a lot of polarity lipids, let's call it like this, accumulating right underneath. So the point where this infectious thread will hit the basal membrane. Now, what you can see here already is that there is, of course, the little space. There's the two membranes or the two cells attaching to each other. And we were interested in what is happening exactly here when this infection thread hits the target. So just to show you a scanning EM image, this is one of these infection threads. It goes from one cell to the others. Here it's broken open. There's a bacteria inside. And we were interested in what happens on the cell-to-cell interface, on this what we call a transcellular passage site. For this, we established a method, and that was mainly done work by Marta in the lab, who runs the EM. And this is correlative microscopy. So we basically combine now fluorescent microscopy with electron microscopy. And the beauty of it is, of course, your signal, like you see it here, it gets blurry at some point. It's resolution limited. But then you have the full-blown depth to look for morphological differences when you look with the EM on it. So the CLEM allows you to find exactly the site back with the electron microscope. And this is a fluorescent labeling of a pectin structure. This is this infection thread again. This is basically part of this one. And here is the basal membrane. And you see that there's also a patch lighting up. Yeah, and when we now look into with EM into it, we find it's exactly, of course, where the cell wall is, but we can also measure now that there's a cleft opening. Basically, the infection thread comes and a cleft opens, just to show you that you gain, of course, a tremendous resolution. So the question for us now is, can we couple FLIM data with morphological data? Yeah, and as an example, for example, I want to show you something. This is a receptor interaction. This is classical FLIM FRET analysis, right? Where you see, there's some dots, Joe. Now it's for you. Yeah, there is some interactions in these nanodomains. Yeah, so for this, there's some interaction in these nanodomains. Here we go. This is highlighted in blue here. And the question for us is, for example, in one of the projects, where these receptors and other proteins interact, is there also a morphological change of the membrane? And of course, there's no way we see this in a low scale with confocal imaging, given the resolution. And in the end, we would only have pixels here. So the question for us is there a membrane invagination underlying this? So for this, we would basically need correlative microscopy to then say, we know there's a specific position in the cell where receptor proteins interact. And can we see if there's a morphological structure underlying this, a cell morphology structure underlying this in these sites? So this is something where we try to basically do FLIM-CLEM to combine this really with protein interaction data and the morphological data. And the other thing that we're pushing a bit more in the future is that people talk about condensates in plant cells and other cells. There's a controversial concept. I think there is some beauty behind it. However, I think to show this in vivo and convincingly that those structures are important and relevant still remains open. So we want to target using FLIM those structures a bit more, and really to see and measure viscosity, for example, of those structures in a cell, and see whether, for example, if we have an invagination like here, and we have data indicating that there is a condensate forming underneath this interacting receptor complex. So do we see this condensate? Can we measure it? We will play around again with rotary probes. I don't know if Joris is in the audience, but so there's a lot of development in this. There's a lot of also tricky parts with the rotor probes, but I think there's, when you look also in the animal feed, a lot of potential. So I think FLIM can help there all to really resolve it, go from protein interactions, getting some cool data on morphology changes or even dynamic changes, for example, with viscosity. And with this, I would like to finish the talk. I hope I'm okay in time. I would really like to thank the lab and really acknowledge Bear here, who again should have given the talk. I hope I didn't disappoint you too much, Bear, in the way I did it. And the team, which is a marvelous team for all the support. I want to also point out Marta's work on CLEM. So establishing CLEM in our lab, and then past members who contributed to the FLIM work, collaborators here in Freiburg, and really the people from the imaging center that helped us a lot. And I have to say also that Leica was very generous, providing us with some support after Bea has been teaching in a Leica course there. So thanks very much for the attention, and I'm open for questions. Thank you.
Wow. Thank you very much, Beatrice and Thomas. That was a fantastic talk. We've just bought a fancy spinning disc and had it delivered, and now I'm thinking maybe we should have got a lifetime system. Anyway, so yeah, the talk worked. So we'll go to a few quick questions. So Yanis has asked, so just to say to the audience, if you populate the question box with your questions, you can do it during the talk as well, and then we'll be able to ask them all at the end. So he's said, very fascinating, Thomas and Beatrice, which I totally agree with. I was wondering something very technical. Are your images acquired with tau sense? And if yes, which mode? Is the phaser analysis on a paid version of LASX?
Yeah.
Yeah, I guess that was for me. No, it's a not thousands. It's actually with a Falcon SP8, so it's having a full FLIM capability or analysis, and the phaser. So I mean, the phaser comes with a license, with a Leica license, right? There's offline, so there's softwares that are available, alternative, let's say, to the one native from Leica, which do phaser analysis, but the tools that Thomas has shown, indeed, they are all belonging to the specific Leica software, indeed.
Brilliant.
Hope that answers. Yeah.
Yeah, that's fantastic. And there's another slightly technical question from Michaela. So thank you for a great talk. Agreed. If multiple lifetime components are present but don't form clearly resolvable centroids on the phaser plot, is there a mathematical approach to extract their fractional contributions, for example, through linear unmixing using known reference lifetimes?
I should take it, right?
Yes, take it, yeah.
I think there's this. So what Thomas showed, there's a tool again in the software from Leica, but I think that would also be available in some of the softwares that are available free of license. That it's indeed able to calculate the fractional contribution, but you need to know the position, so the GNS coordinates of the two species that are basically mixed together at the site where you wanna resolve their intensity contributions. And I mean, mathematically, and this would be with a phaser. Otherwise, of course, you can do fitting. But as Thomas said, I think the phaser is really much more accurate, especially at low photon counts.
I'm going to take chair's privilege and ask one quick question. So, and then we'll move on for time's sake. How quickly does it take to acquire an image? So, what kind of frames per second do these? Because presumably they take a bit longer than a normal imaging system, because obviously you're doing all the photon counting.
I think, well, we have, let's say, it depends how many accumulations. So you want to have, basically, for FLIM, it's important that you get quite some photons, right, from your image. So depending on how strong your signal is, you have to choose basically how many times you want to scan a focal plane. And that basically tells you a little bit of time that you're gonna take to acquire an image, right? If you want to, so normally, let's say it can be from 1 second, right? It's gonna, just the time to scan, or longer. It depends.
Okay. And I think this is really the point where I think there's a limit when you're talking about moving particles. So as soon as the structures are moving and lowly fluorescence, then there is a limitation. Yeah, it's probably something to solve over time now, to really use tracking modes to keep this, but I think that is a challenge still.
Yeah. Okay.
Just that I
Well,
also answered a question, or at least thanks for joining.
Well, that was a wonderful talk. We're going to move on, but I just want to make one very brief point. There's lots of very technical questions in the question session, which I'm taking as a sign that this seminar series is working, which is really good. So people are engaged and asking very good questions, which is also a testament to your work, Beatrice, and Thomas as well. So on that note, I say thank you very much again, and we're going to move on now. So thank you.